There are two types of control mechanisms associated with digestion. They are neural control and hormonal control. Let's have a look at the hormonal control mechanisms of digestion. 5 different hormones are associated with hormonal control mechanism. They are GASTRIN, CHOLECYSTOKININ, SECRETIN, glucose-dependent insulinotropic peptide or GIP, and motilin. Now we can see all these one by one. Gastrin is secreted by G cells of antrum region of stomach. Secretion of gastrin is activated by the presence of proteins in the stomach, distension of gut, and by the activity of gastrin releasing peptide released by the neurons in the gut. Gastrin stimulates the secretion of gastric acids and the growth of mucosa. Next is the cholecystokinin, which is secreted by the eye cells of mucosa of duodenum, jejunum, and ileum. Its secretion is stimulated by the presence of proteins, fats, and monoglycerides in the intestine. Cholecystokinin stimulates the secretion of pancreatic enzymes and bicarbonates. This hormone stimulates the contraction of gallbladder and thereby releases bile into duodenum. As you know, bile is important in the digestion and absorption of fats. This hormone also stimulates the growth of pancreatic cells. Cholecystokinin has some inhibitory actions also. They inhibit the secretion of acids in stomach. They also inhibit the contraction of stomach. This in turn slows down the emptying of stomach into small intestine and provides adequate time for digestion of fats in small intestine. They also inhibit appetite and helps to prevent overeating. Secretin is the first gastrointestinal hormone discovered by the humans. It is secreted by the S cells of duodenum, jejunum, and ileum. The hormone is secreted in response to acidic gastric juice entering the duodenum and also by the presence of fats. This hormone induces the secretion of pepsin, pancreatic bicarbonates, and biliary bicarbonates. It also reduces gastrointestinal mobility and promotes the contraction of gallbladder. Secretin also promotes the growth of pancreatic cells. Apart from all these stimulatory effects, secretin also inhibits the secretion of acids from the stomach. The inhibition of gastric acid secretion and release of bicarbonates reduces the acidity in small intestine. Glucose-dependent insulinotropic peptide or GIP is secreted by the K cells of duodenum and jejunum. It is secreted by the presence of fatty acids and amino acids. Carbohydrates also show mild influence in its secretion. Primary activity of GIP is the stimulation of insulin secretion. It also inhibits gastric acid secretion and decreases motor activity of stomach. This is another hormone that helps in slowing down the emptying of stomach contents when there is food materials in the upper region of small intestine. The final hormone, motilin, is secreted by the M cells of stomach and upper duodenum only during fasting. It's activated by fasting. Motilin increases the motility of gastrointestinal tract. This hormone is released cyclically and it creates cyclic intestinal waves passing from stomach to small intestine. The secretion of motilin is inhibited by the ingestion of food materials. That's all about hormonal control.